Hi, my name's Laura. I'm a biologist with the Immune Tolerance Network, and today I'm going to be demonstrating the urine processing procedure. If you're doing this for the Vibrant trial, this is known as urine processing procedure A, but procedures B and C are very similar. So let's get started.
Let's start by looking at an overview of the urine processing procedure. First, the urine sample will be centrifuged to separate the cell pellet from the supernatant. Next, the supernatant will be split into two different products, one with added protease inhibitor and one without, which we call neat. Ultimately, you will be cryopreserving three things: the two types of supernatants, with and without protease inhibitor, as well as the cell pellet. For this procedure, you should start with a minimum of 15 milliliters and a maximum of 90 milliliters of urine. First, swirl the cup to resuspend any particles that may have dropped to the bottom. Next, pour the urine into two 50 ml conical vials with half of the sample in each vial. And now I'm ready to take the samples to the centrifuge. Now that my tubes have come out of the centrifuge, I'm going to separate the supernatant from the cell pellet by transferring the liquid to a new conical vial. As I near the bottom of the tube, I'm going to slow down and be careful not to disturb the cell pellet. It's important to keep the urine sample, the cell pellets, and the supernatants on ice throughout the entire procedure. And now I'm going to repeat that exact same process with the other tube. The next step is to continue processing the supernatant. First, I'll need 10 cryovials, which should all be labeled neat. Then I'm simply going to transfer 1 milliliter of supernatant directly into each cryovial. Here I'm only demonstrating 3, but once all 10 are filled, be sure to close the caps tightly, and then they're ready to go to the -80 freezer. Now for the supernatant treated with protease inhibitor, I'm going to start by transferring 11 milliliters of the urine supernatant to a 15 milliliter conical vial. The protease inhibitors are supplied as tablets in foil packs, so all you have to do is pop one tablet directly into your tube. Then you can shake the tube vigorously or vortex it until the tablet is dissolved. You should have 10 cryovials labeled protease, so you can transfer 1 milliliter to each vial just as we did before. Again, I'm only demonstrating 3 tubes, but once you've filled all 10, you're ready to take them to the -80. Now we're ready to move on to processing the urine cell pellet. You should have ice-cold ex vivo 10 buffer and 2 cryovials labeled pellet. First, I'm going to resuspend one of the cell pellets in 0.5 milliliter of cold ex vivo 10. To do that, I'm going to pipette up and down, making sure to rinse down the sides of the tube. And then I'm going to transfer all of this liquid to my cryovial. To make sure I don't leave any cells behind, I'm going to rinse the same conical tube a second time. Again with 0.5 milliliter of cold ex vivo 10. And add it to the same cryovial. Now that conical tube can be discarded, and I'm going to repeat the process with the other cell pellet in the other cryovial. I'm only demonstrating one of 2 tubes here, but once both pellets are transferred, you're ready to centrifuge the cryovials. My cryovials are back from the centrifuge and the cells should be pelleted. Next, I'm going to use a disposable transfer pipette to remove as much supernatant as possible without disturbing the cell pellet. To resuspend the cells, I'm going to use ice-cold CryoStore buffer. To do that, I'll add 250 microliters, or 0.25 milliliters, of cold CryoStore and pipette up and down. Once both cell pellets have been resuspended, I'll transfer them to a cold Mr. Frosty container and take it to the -80 freezer.
All right, you've reached the end of the urine processing procedure, so the cell pellet and those supernatants are all in the freezer. Thanks for all your hard work, and good luck.